Welcome to Basic Biochemistry for Creationists who are non-biologists and non-chemists. Even though this is only the fourth episode in the series, we have almost enough biochemistry to address some tremendous topics related to creation, evolution, abiogenesis, and intelligent design. So I thank all the participants who've patiently gone through the first three lessons. God willing, maybe somewhere in the next few episodes, we'll start using biochemistry to see evidence of miraculous designs in biology. However, in the meantime, we still have some more mundane things to learn before we can really get going, so I thank you all in advance for your forbearance. Here is where we left off in the last episode. Since the previous episode will be referenced frequently in this present video, it may be helpful to review the previous 9-minute episode as needed. After all, it's only 9 minutes long. The top of the diagram represents segments of three identical primary structures of collagen, but colored differently for clarity so as to correspond to the three identical but differently colored collagen polypeptides intertwined together at the bottom. I'll refer to this diagram again to introduce the concept of protein quaternary structure, but before getting to quaternary structure in detail, we need some preliminary concepts and terms. As covered in episode 3, a polypeptide is a polymer chain made of amino acids analogous to a necklace where each bead on the necklace represents an amino acid monomer. Each protein is made of one or more polypeptides. What can lead to some confusion is that it is not always immediately clear when reading scientific literature if the protein in question is made of a single polypeptide or of multiple polypeptides. The singular word polypeptide at least indicates when we are talking specifically about a single polymer chain of amino acids, whereas the plural word polypeptides indicates we are talking about multiple polymer chains of amino acids. Also, just to emphasize again, a polypeptide is a special case of a polymer. So, we understand what a polymer is, right? Right. But, and this is a big but, a big, huge, Kim Kardashian-sized but. And the but is that the meaning of the word monomer can be a source of confusion. Though a polymer, such as a polypeptide, is made of monomers of amino acids, an entire polymer itself, specifically a polypeptide, when part of a larger molecular complex, can also itself be called a monomer, and this can cause some confusion. I repeat, though a polymer such as a polypeptide is made of monomers of amino acids, an entire polymer itself, specifically a polypeptide, when part of a larger molecular complex, can also itself be called a monomer. To hopefully alleviate the confusion, let me give a little example using the collagen protein we discussed earlier. A typical functioning collagen protein is composed of three identical polypeptides of collagen. An individual polypeptide in this complex of three polypeptides, such as the green one, is called a monomer. I personally prefer not to use the word monomer to describe a single polypeptide in order to avoid confusion, but I'm pointing this out in case you encounter someone using a different convention than the one that I prefer to use. Not surprisingly, the prefix mono in the word monomer corresponds to the number 1, the number of polypeptides under consideration. And if hypothetically we had two polypeptides connected together to make a complex, we call the complex a dimer, where the prefix di in the word dimer corresponds to the number 2 for the two polypeptides in the complex. Likewise, when we have three polypeptides connected together, we call it a trimer, where the prefix tri in the word trimer corresponds to the number 3 for the three polypeptides in the complex. For this trimer, some will say the individual polypeptides are monomers, but as I said, I don't like using that terminology. I prefer to simply say the individual components of the trimer are polypeptides, not monomers. The moral of the story is the word monomer has different meanings depending on the context. In one case, the word monomer can refer to individual amino acids within a polypeptide. In another context, the word monomer refers to the entire polypeptide itself when the polypeptide is part of a larger molecular complex, like a protein such as the collagen trimer shown here. Although it is not incorrect to call a single collagen polypeptide a protein, you'll notice in episode 3 I tended to call a single collagen polypeptide a polypeptide rather than a protein. That's because in many cases a functional collagen protein is a trimer of collagen polypeptides. By the way, the actual collagen polypeptides are much longer than those depicted here. I'm only showing a small segment for simplicity. Also, in episode 3, we learned a polypeptide's primary structure is its amino acid sequence. The next easiest to understand structure of a protein is its quaternary structure. The secondary and tertiary structures are a little harder to understand, so I'm saving those for another time. So what is quaternary structure? Quaternary structure is a structural description of the characteristics of a protein according to a checklist that looks something like this. Quaternary structure A describes the number of polypeptides in a protein, B describes whether the polypeptides are identical or not, and C describes the arrangement of the polypeptides provided such an arrangement is actually describable in words. I'll use some shorthand for the checklist so we can fill in the blanks later. So let's try to fill in the blanks in the checklist for the protein collagen based on the data gathered and reported by protein researchers. A. The number of polypeptides is 3. B. The polypeptides are all identical, so yes. And C. For arrangement, the polypeptides are intertwined into a helical arrangement, so helical for C. That wasn't so hard, was it? It's not too hard to describe the quaternary structure as long as you're not the one actually having to do experiments to determine the actual data required for this checklist. But if you're the one in the lab whose job it is to gather such data, ouch! Since such tasks in days gone by occupied the staff of an entire laboratory working years to determine that data. Returning back to our filled-out checklist for collagen, there is a more compact way to describe the data in this checklist with a short phrase such as helical homo trimer. There might be other phrases that are practically equivalent to this phrase, but that's the phrase I'll use for our purposes today. Let's see how I constructed this compact phrase. Because the polypeptides are arranged geometrically as a helix, I use the word helical. Because all the polypeptides in this collagen protein are identical, I use the word homo. Now, if hypothetically the polypeptides were different rather than identical, I would have used the word hetero. But since they are identical, I used the word homo. For proteins made of 3 polypeptides, I use the word trimer, where the prefix tri means 3. By way of extension, if, hypothetically, the number of polypeptides had been 4, I'd have used the word tetramer, if 5, pentamer, etc., as shown in this table. Well, that's just about it for this episode. We're hopefully now acquainted with some basics of biochemistry, such as protein primary and quaternary structure. And we now have a good base of knowledge to begin studying arguments in favor of intelligent design and miraculous creation of life. God willing, I hope to explore some of these arguments in subsequent episodes. So thank you again for joining us in yet another exciting episode of Biochemistry for Creationists. God bless.